Morning students. I am today going to teach you the remaining part of the innate immunity, and in this the next thing I have to tell you is about the receptors of innate immunity. The receptors of innate immunity, the antigenic substances display a regular molecular pattern. Those on pathogenic microbes show PAMP, means pathogen associated molecular pattern, and the antigenic substance on damaged cells show DAMP, that means damage associated molecular pattern. Means to say that each molecule has a definite pattern by which that antigen can be recognized. Now these patterns are recognized by pattern recognition receptors present on surface of cells of innate immunity. Okay, now we shall see what are the types of the receptors. The types of the receptors are: number one, plasma membrane receptor, and this is to detect extracellular microbes. Next type is the endosomal receptor, and this is to detect the microbes ingested in endosomes, about which you have been taught earlier. And the third one is the cytosolic receptors, which is to detect the microbes in cytosol. Now come to the classes of the receptors. There are 6 classes of receptors has been described. You can see TLRs, NLRs, CLRs, RLRs, GPCR and Mannose receptor. One by one I will deal with all the receptors. First one is the TLR, means toll-like receptors. Toll-like receptors, it is the founding member, name has been given on the founding member toll, and it is first discovered in Drosophila insect, about which you must have learned in your genetics during biology classes. Now there are 10 TLRs, 10 toll-like receptors, each recognize a different set of microbial molecules. For example, TLR4 recognize LPS, means lipopolysaccharide on gram-negative bacteria. TLRs are plasma membrane and endosomal receptors, which on binding with antigens transduce signal pathway culminating into a common activation of 2 sets of transcription factors. Those transcription factors are NFKB. It causes synthesis and secretion of cytokines and expression of adhesion molecules. Both these result into causing recruitment and adhesion of leukocytes. Now next transcription factor is IRFs, means interferon regulating factors. IRF means interferon regulating factors. It is the name of the transcription factor. It stimulates the production of antiviral cytokines, that is type 1 interferon. Now come to another type of the receptor, which is known as CLRs, means C-type lectin receptors. These are the extracellular receptors present on plasma membrane of macrophages and dendritic cells, about which you have been told earlier. And it is to detect fungal polysaccharides, and that results into, after interaction, it results into inflammation against the fungi. Now next type of the receptor is RLR receptor, that is rig-like receptors. It is a cytosolic receptor in most cell types, which is present in most of the cell types, and it is to detect nucleic acids of viruses that replicate in cytoplasm of infected cell, and after interaction with this receptor, antigen and receptor, it results into the antiviral cytokines production. Now, next receptor is the GPCRs, that is G protein coupled receptor. This is again extracellular receptor, and it is present on leukocytes, for example, neutrophil and macrophages cell membrane, and it recognizes the short bacterial peptide containing N-formyl methionine residues. And after the recognition and interaction, it results into the chemotactic response to infecting bacteria. The chemotaxis, you already know, is done by the neutrophils and the monocytes, and these go into the infected area to deal with the bacteria. Now, next receptor is the Mannose receptors. Mannose receptors are again extracellular receptors. They recognize the terminal mannose residues present on microbes, and after the interaction, it results into the phagocytosis of the antigenic molecule or the microbe. Now, the sixth receptor is NLR, that is node-like receptors. Node-like receptor, the founding member of this group is the node 2. Node means nucleotide oligomerization domain. Nucleotide oligomerization domain, that is a full form of the node. Now, what does it do? It recognizes a wide variety of substances, including what you can see here, present in the cytosol and present in the on the cell membrane and also in the endosome. All the antigens present at all these sites, all the three sites, they are recognized by this receptor. Now, you can see how many types of the receptors of the NLR receptors, that is cytosolic, that is endosomal, and that is extracellular. So, first we shall deal with the major one, that is cytosolic receptor 1, that is node-like receptor protein. Now, you see here first that what are the stimulus, what are the substances to which this node-like receptor respond. You see here, it is pathogenic microbes and, for example, bacteria and their products. Next one is extracellular necrotic cell products, which contain, for example, ATP. These things stimulate this receptor, and you can see it has stimulated the node-like receptor protein. This is node-like receptor protein. It is the cytosolic protein, and this cytosolic protein 3, node-like receptor protein 3, is rich in the leucine. Here you can see this is the node-like receptor protein. Now, there is the adapter protein here, shown diagrammatically, and there is inactive caspase 1, shown diagrammatically here. So once there is stimulation of this one, that is another point of stimulation, so is the lower concentration of the potassium inside the cytosol, which is produced by the efflux of the potassium outside the cell, reactive oxygen substance, and presence of crystals. Crystals may be urate crystal, it may be lipid crystal, or it may be cholesterol crystal, which I'll see later. So all these things stimulate this one. They act with this node-like receptor protein, and it results into binding of these three things, that is this NLRP3, adapter protein, and inactive caspase. You can see here how these has interacted. So this thing, which has been formed by the combination of all these three, is a multi-protein complex known as inflammasome. Now this inflammasome, you can see the result of formation of this inflammasome is the activation of this inactive caspase 1. You can see here is the active caspase. Now this active caspase has two roles. One is you have already known earlier that it is responsible for production of pyroptosis, and a type of the cell death. And another role is that this active caspase 1 cleaves the pro-interleukin 1 beta into active interleukin 1 beta. Pro-interleukin 1 beta is inactive form of this interleukin, and it is cleaved enzymatically, proteolytically it is cleaved into active interleukin 1 beta, which is responsible for production of inflammation. Now you can see the inflammation may be produced by the presence of the urate crystal, which gives rise to the gout, the presence of the lipid crystal, lipid granules, which gives rise to the type 2 diabetes mellitus, and cholesterol crystal, which results into the production of the atherosclerosis. In all these processes, the inflammation is induced. Now you can see here another picture in which you can see how pro-interleukin 1 beta is produced. It is produced by the stimulation by endosomal receptor. Here you can see toll-like receptor is also endosomal receptor. This is endosome which ingests the virus inside the cell, and you can see the nucleic acid of ingested bacteria and viruses. Here you can see of bacteria. Now this nucleic acid of the ingested bacteria present inside this endosome stimulate the bind to the toll-like receptor here. That is the endosomal receptor. Now this, after being activated, the toll-like receptor transduce the message to the nucleus of the cell, the host cell, and there it stimulates the process of transcription, by which there is the production of pro-interleukin 1 beta. This is the whole story about it. Now you can see here the other types of the receptors about which just I have discussed. Rig-like receptors about which we have discussed. This is again a cytosolic receptor, and it interacts with the viral RNA, and then it produces certain message to the nucleus. Another extracellular receptors are GPCR. You can see here there is 7 molecule of the G peptide, G protein associated receptor. See GPCR, G protein. Here you can see G protein coupled receptor. This is here the G protein coupled receptor, and this is also extracellular receptor. Another extracellular receptor is the Mannose receptor, and there is the C-reactive lectin, C-type lectin receptor. This is C-type lectin receptor. You can see here the C-type lectin receptor binds the fungal polysaccharides, while others are binding the other extracellular molecules. So these are all about the receptors. Now one thing you have to understand, that the gain of function mutation in NLR results into periodic fever, which is also known as autoinflammatory syndrome. Now you will know about the gain of function mutation while we shall discuss the genetics. Now come to, after finishing the receptors of the innate immunity, come to the mechanism of innate immunity. It is the innate immunity provide early host defense against microbes as below. Number 1, it is by induction of inflammatory reactions. How it is induced? By producing cytokines, complements, other mediators, and which cause the initiation to provide early host defense against the microbes as below. Number 1 is the induction of inflammatory reaction. It is by producing the cytokines, complements, and other mediators which cause initiation of vascular and cellular components of inflammation, and it results into the recruitment of leukocytes to destroy microbes and to ingest and eliminate the damaged cells. These are number one. Number two is the production of antiviral defense, that is viral antigens and inside the infected and outside uninfected cell, that is that results into production of the interferon, and it causes activation of the enzymes. These enzymes degrade viral nucleic acids and inhibit the viral replication. Third thing is, and most important, is the stimulation of the subsequent more powerful and more sustained adaptive immune response. That's all for today. Thank you.